Hi, everybody, and welcome to the third three-piece session. This is brought to you through a collaboration between the Cure Parkinson's Trust, World Parkinson's Coalition, and the Van Andel Institute. Today, we have three speakers presenting on the theme of membrane trafficking in Parkinson's disease, with these results all generated from Mark Cooksen's lab at the NIH. The session will begin with Luis Bonet-Ponce, that will give an introduction to the theme and present on uncovering the role of LRRK2 in the lysosomal membrane. And then we'll next have Adamantios Mamais, who will be presenting on LRRK2 modulating iron uptake in microglia. After the second presentation, Lisa will then introduce the third speaker, and then we'll finish with an overall Q&A session. So if you look at the bottom of your screen, there's the Q&A feature where you can add questions throughout whenever you think of the question. And if you've got a question for a specific person, please include their name at the beginning of your question so we can direct it to the correct person at the Q&A portion of the session. Lisa, if you want to introduce yourself, we can just get started. That's fine. Sorry, Lisa is the co-chair for today's session.
Hi, everybody.
My bad. Okay, so we'll now pass over to Louis, if you would like to start sharing your screen and begin the presentation.
Yes. Hold on. Okay. Is it good? Yeah. Okay. So, hi, my name is Luis. I work as a postdoc at the NIH, and today we're going to talk about LRRK2 and lysosomes. I would start thanking the organizers, Lisa, Michaela, Hella, and Eros. I think they've done an amazing job with this. I really love, every Tuesday and Thursday doing this, and I'm excited for today's talk. So, LRRK2 mutations in the LRRK2 gene cause familial Parkinson's disease, and we also know that variation in its locus increase the risk to develop the sporadic form of the disease. At the protein level, LRRK2 is a very large protein, and we know that has two catalytic domains. So first we have the ROCOR by domain that has GTPase activity, and the kinase domain with kinase activity. The other three domains are proposed to play a role in protein-protein interaction. If we look at the pathogenic mutations, all of them are clustered in these two catalytic domains. They are gain-of-function mutations. And interestingly enough, mutations in the ROCOR domain somehow also affect kinase activity. So the overall result is a toxic hyperactive kinase protein. However, how this toxic hyperactive protein leads to PD is not known. And even if we go to the more basic question, which is what does LRRK2 do in the cell, that's still fairly elusive. So that's what drew me to study this protein when I joined Mark's lab. What we know is that LRRK2 is able to phosphorylate a subset of 14 RAB TPases, which basically puts LRRK2 in the context of membrane trafficking. Now, when I joined the lab as well, I wanted to focus on the lysosome. And the reason why is because we know, and it's been pre-accepted at this point, that lysosomal biology is a key aspect in PD pathology. Now, the link between LRRK2 and the lysosome is not new. Actually, we know that cells that carry LRRK2 mutations present lysosomal dysfunction. Now, whether those are direct or indirect through LRRK2, it's something that is not known. So that was the main goal of this project, is to try to understand a specific function of LRRK2 in the lysosomal membrane. So for that, what I first did is to move away from cancer cells, because lysosomal biology is really different for obvious reasons. And so when we look at the cells, primary cells from the mouse brain, and we look at LRRK2 expression, mouse primary astrocytes are the cell type that expresses LRRK2 to the highest level. So we decided to start using mouse primary astrocytes for this project. First thing that we did is to express LRRK2 in the cells. So half of the cells present this scenario, which is the most reported one, that LRRK2 seems to be diffused in the cytosolic membrane, and the cytosol, sorry. This is what you see in neurons, this is what you see in fibroblasts, and in all cancer cells. However, in mouse astrocytes, the other half of the population all of a sudden is able to recruit LRRK2 to a subset of lysosomes. This is just a very small subset. We're talking about maybe 3 lysosomes per cell. So it seems to be like a very specific population. So to try to understand more of what LRRK2 might be doing there, we did the proteomics assay. So basically the idea here is to tag LRRK2 to an APEX2 sequence. And what APEX2 does is, in the presence of biotin phenol and hydrogen peroxide, is able to covalently bind biotin to every protein that is within 10 nanometers. So you combine this with an APEX2 bind to a nuclear exporting signal to eliminate the cytosol. In this case, you basically enrich for membrane proteins. So we did that in a mass spec, and we actually found a lot of membrane proteins, which is good. Then we selected the ones that have a known lysosomal function, and we clone them in a GFP construct and expresses with LRRK2 in astrocytes. And we found that only GIP4 was able to colocalize with LRRK2 positive lysosomes. GIP4 is a member of the GIP family. And what it does basically is gets recruited through small GTPases and then to a membrane, and is able to bind to motor proteins, to dynein and kinesins, and then promote movement of those membranes. Now, if you look at the lysosomes here, LRRK2 positive lysosomes are enlarged, which is a sign of damage. And if we look at the staining of the lysosomal enzyme cathepsin B, you will see how the levels of cathepsin B are very, very low, or sometimes in the yellow arrows, almost non-detectable, which lead us to think that maybe the lysosomal membrane in these lysosomes may be broken, allowing cathepsin B to leak outside the lysosome. So to test that hypothesis, what we did is to treat cells with a compound called LLOME. So when you put LLOME to the cells uptake the drug, the compound, and when it moves to the lysosomes, it gets condensated by cathepsin C, then leading to lysosomal membrane damage. So we tested that in our astrocytes, and we found that the presence of this drug, LLOME, leads to a very dramatic increase in LRRK2 positive lysosomes, thus suggesting that LRRK2 indeed goes to rupture lysosomes. Now the cells, what it does, when lysosomes are broken, it initiates this process of eliminating them through a specific type of autophagy called lysophagy. The way, oh, sorry. I don't know what I'm doing. Okay, so the way the cells does that is by recruiting galectin-3, which is a marker of lysophagy, and then initiating the whole process. So to try to see whether LRRK2 may be playing a role in lysophagy, we stain them with galectin-3 and inhibit LRRK2 kinase activity using MLI2. When we did that, we did see no difference in galectin-3. Now, you may argue that maybe LRRK2 is affecting something downstream of galectin-3. So we look at the expression of LC32. And we saw that in the presence of LLOME, when we inhibit LRRK2 kinase activity, we still don't see any difference in LC3 expression, meaning that when we rupture the lysosomes, LRRK2 is playing an independent role to lysophagy. Okay, so go back to GIP4. We see that when we add LLOME, GIP4 also increases presence in the lysosomal membrane. However, the spike of expression takes longer than LRRK2. Basically, you see how GIP4 gets recruited, especially at 6 hours, when LRRK2 happens in 2 to 4 hours. And also, if you look at the blue arrows, there is several LRRK2 positive that are GIP4 negative lysosomes, which basically allow us to believe that LRRK2 may come first and then recruit GIP4. And that's what happens. When we inhibit LRRK2 kinase activity, we see no evidence of any recruitment of GIP4 to the lysosomal membrane, which allows us to think that is LRRK2 kinase activity dependent. Now, this is with overexpressed LRRK2 and also using a compound that might be unspecific. So we validated that also using LRRK2 knockout astrocytes, and we can see that the whole recruitment of GIP4 is completely dependent on LRRK2. Now, when we look at 2 pathogenic mutations, we see that the mutation, the G209S mutation that is in the kinase domain, heavily increases GIP4 localization, which basically means that it is kinase activity dependent. Now, there is this gap between LRRK2 and GIP4 that allows us to believe that maybe other proteins that are actually bridging LRRK2 and GIP4. So to look for them, we repeated the APEX screening, in this case, plus minus LLOME. And we saw in yellow 2 RAB GTPases that are known substrates of LRRK2. Those are RAB10 and RAB35. And both of them also go to LRRK2 positive lysosomes when we treat the cells with LLOME. And also, as happens with G4, the recruitment of these proteins to a lysosome is completely dependent on LRRK2. Now, the hypothesis that we have then, as I said in the introduction, G4 goes to membranes through small TDPases. So our hypothesis is that LRRK2 goes to lysosomes, recruits the RAB, phosphorylates the RABs, and then G4 goes there. So we checked whether that was true, and we saw that using a phosphate gel for RAB35, that in fact, LRRK2 in the presence of LLOME is able to phosphorylate RAB35. And the same happens with RAB10. So to make the link between the RABs and G4, we created the phosphonull construct. So instead of having the threonine 7273 side, we'll have an alanine, preventing the phosphorylation, right? And so to check whether the phosphonull constructs are able to recruit G4. When we do that, if we prevent the phosphorylation by LRRK2 of the RABs, G4 is completely unable to go to the lysosomal membrane. So basically, we have the system where LRRK2 phosphorylates the RABs and recruits them, and that phosphorylation is required to then bring G4 to the lysosomal membrane. So basically, G4 acts as an effector in this context. But what does it mean for the lysosome? So we did super-resolution microscopy, and we found that several of these lysosomes are able to tubulate a G4 tubule out of the lysosome, and that tubule is negative for LRRK2 and negative for LAM1. This is a max projection, so you can see it really well. So this is more clear. This is a 3D reconstruction, and you could clearly see how the tubule is negative for LAM1. Now, this is really strange, because if it's come from the same membrane, how come the lysosome is able to prevent LAM1 to go to the tubule if it's the same membrane? So to make sure that it is actually a continuous membrane, we did FIB-SIM microscopy. And you could clearly see here that it is from the same membrane, but somehow the lysosome is able to prevent LAM1 to go to the tubule. Now, because this is FIB-SIM, we can image every 6 nanometers. So we did a reconstruction of the lysosome, and we could see two cool things here. One is the ER is able to grab the base of the tubule, and the other, more interesting for our story, is the presence of a microtubule in the tip of the tubule. Now, if you remember, G4 is able to bind to membranes and then bridge those membranes to microtubules, right? So our hypothesis now is that G4 is probably doing, by binding to motor proteins and microtubules, elongating the tubule to form a tubule. So to test that hypothesis, we first needed another marker that was at the membrane, at the tubule, sorry. And the only one that we found was endogenous RAB10. RAB35 was not at the tubule, and any of the other typical lysosomal markers were on the membrane as well, at the tubule, sorry. So what we did is to knock down G4 and look at for RAB10 tubulation, and we see that when we depleted G4 expression, there is a decrease in tubules. Therefore, meaning that that's the role of G4, forming the microtubules, forming the tubules, sorry, probably through microtubules. Now, there's something interesting also in these tubules. They are very heterogeneous. So this is a FIB-SIM reconstruction. You will see this is a one lysosome forming 5 different tubules, and you will see how different they are. They have different shape, different morphology, they have different thickness and different length. For the most part, the longer the tubule is, the thinner it is, but it's not always like that. So it's something really odd and specific to these tubules that will be really cool to monitor moving forward. Okay, now this really reminds me to what you see in early endosomes, which is when early endosomes need to recycle cargo to either the plasma membrane or the trans-Golgi network, they form these tubules, then there are scission vesicles, they move to these different compartments. However, lysosomal sorting is not a mechanism, a process that has been discovered yet. In theory, lysosomes just uptake things and degrade them. They don't sort things or vesicles outside of their own compartment. So to make sure that this was actually lysosomal sorting, we did live cell imaging with super-resolution microscopy. So you will see how a bunch of lysosomes will form a tubule that will then scission and form vesicles that will move around the cytosol. This second move is a little bit better. You'll see how a tubule is formed, and then vesicles will be moved and moving around the cytosol. So basically, trying to understand the basic biology of LRRK2, we stumble into a new cellular process that sorts vesicles outside of the lysosome. So we call this process LITTLE, that stands for lysosomal tubulation and sorting driven by LRRK2. Okay, so this is very cute, but where do these vesicles go? We never see them going out the plasma membrane, and we never see fusion of these vesicles to other compartments. What we do see is these vesicles being attached to other lysosomes. So here you could see in the EM level that there is a membrane contact site of these vesicles. And here's another example where you could see a moving vesicle interacting with a lysosome, staying there for a little bit, and then moving elsewhere. So it really seems like these vesicles are able to form a transitionary contact site with other lysosomes, probably healthy lysosomes, and interchange something there that we haven't gotten there yet, but that's something that I'm very, very passionate about. So to summarize, we have that once a lysosome has the membrane is ruptured, instead of getting degraded by lysophagy, recruits LRRK2 and promotes this cascade of events that leads to a tubulation and sorting of vesicles that interact with other lysosomes. This is kinase activity dependent and is based on the phosphorylation of two RAB GTPases, RAB10 and RAB35, and then recruit the modulating protein G4 that through microtubules forms that tube. I would like to thank a lot of people in this project. I don't have time to go one by one. And also, if you guys are interested, this paper is under review now, but you can find it in my archive. And with that, we'll go with my good friend, Adam. If I can unshare my screen, which I cannot do right now. So, all right, hold on. Have I done it?
Yeah, I think so.
Okay, cool.
That was very nice talk. That's great. So we were told we should stop our video to increase bandwidth. Can everybody hear me, Gary? So I'm going to stop my video and try to share my screen. Does that work? How do I close? Yeah, does that work? I think so. It must do. Right. So, hi everybody. I want to thank the organizing committee and Lisa and Michaela for organizing this. Right. So my name is Adam. I did my postdoc at the National Institute of on Aging with Mark Cookson. So I'm going to present work that I did there in the last 5 or 6 years. And as you'll see here at the mood working stage, I'm in between jobs right now. So I was working at the NIH and now I'm moving to the lab of Matt Lavoie and Malu Tanzi at the CTR&D in Florida. And this is me mood working myself in a very cool way. So I'm going to talk to you about LRRK2 and the role of LRRK2 in regulating RAB8-mediated recycling in downstream effects of transferrin and iron modulation in microglia. Right. I think that works. Okay, right. So there won't be any introduction because Luis did this very well. But all I'm going to say about LRRK2 is that we believe that LRRK2 is involved in a small number of different functions in different cell types in the brain. So there are, So LRRK2 is expressed in different cells in the brain, and there's a small number of physiological functions that we believe that LRRK2 is involved, namely autophagy, vesicular trafficking, mitophagy, the endolysosomal system, and inflammation. So what we care about and what I've been focusing on is the downstream effects of LRRK2 phosphorylation on RAB GTPases and specifically RAB8. Now, RAB8A is a small RAB GTPase that is involved in recycling of the transferrin receptor, right? Of the recycle. Yep. I can't hear anything. This is going fine, right? I can't actually see my comments or anything. I think, yeah. Wait. Yep, that's fine. Oops. Yep. Okay. Right. So, so I was saying RAB8 is involved in recycling of internalized receptor, and more specifically, we find RAB8 in the endocytic recycling compartment, in recycling tubular endosomes, mediating the return of receptors and cargo after internalization through clathrin-mediated endocytosis back to the plasma membrane. So RAB8 is phosphorylated by LRRK2, and pathogenic mutations of LRRK2 increases phosphorylation, and we're trying to see what the effects are of LRRK2 PD-linked mutations on RAB8 and recycling. So one of the first things to do is to stain for endogenous RAB8 in cells. So this is HEC293 cells, where we stain for endogenous RAB8 in the context of transiently expressing LRRK2 mutations. So what we see here is that in cells expressing wild-type LRRK2, RAB8 is localized in these recycling tubular endosomes, which are not associated with lysosomes, but in cells expressing the R857G or G2019S mutations that reside in the enzymatic domains of LRRK2, we see the sequestration of endogenous RAB8 onto LAMP2 positive lysosomes. And this is a similar phenotype to what Luis has seen with LRRK2 mutations going to the membrane of damaged lysosomes. Now, to further examine whether this is a kinase-dependent effect, we can inhibit LRRK2 by treatment with a specific LRRK2 kinase inhibitor MLI, and we see that we can ameliorate, we can effectively rescue this phenotype, and RAB8 within a couple of hours will return back to the functioning tubular endosomes. A way to quantify this is by high throughput imaging, and in this case, we're looking at the blue bar. That is basically the percentage of cells that have sequestered the RAB8 phenotype. And we can see that all the mutations have the same effect. So we see a convergent phenotype of LRRK2 PD mutations on RAB8 localization, and we can rescue it by kinase inhibition. At the same time, we can quantify the colocalization of RAB8 with lysosomes, and it follows the same profile with the mutations increasing the colocalization of RAB8 with lysosomes. Sorry, these are the lysosomes, and also colocalization with LRRK2, which you can obviously see in the actual images. So to look at the link between this and the phosphorylation status of RAB8, we can look at the phosphostatus of the threonine-72 site by Western blotting. And in this case, it's exactly the same setup where we quantify threonine-72 phosphorylation, and we see an increase with the mutants, which follows basically the same profile with the morphology. So we have a kinase-mediated effect on RAB8 phosphorylation and subsequently on its intracellular localization. Now, if RAB8 is mislocalized away from its physiological compartment, what happens to RAB8 functions? And one of the ways to look at that, oops, no, hold on. That was the next slide. So before I go there, so in an effort to characterize these RAB8 lysosomes a little bit better, RAB8 positive lysosomes a little bit better, now this is going into the realm of Luis's study as well. What we looked at is the cathepsin D content of these LRRK2 positive, RAB8 positive lysosomes. And what we see is that the majority of them are in fact cathepsin D negative. So we believe that we have recruitment of RAB8 into non-functioning lysosomes, into damaged lysosomes. So as I was saying, that to look at the effect of this translocation of RAB8 on its function, one of the ways to look at that is to do a transferrin assay, because RAB8 is involved in recycling of the transferrin receptor and of transferrin back to the plasma membrane. So in this experiment, we're looking at HEC cells again, and we load cells in culture with fluorophore-conjugated transferrin, which is 568 here in red. And after 10 minutes of recycling, we can look at the endogenous localization of this transferrin. So in cells expressing wild-type LRRK2, we see that transferrin has a cytoplasmic localization, presumably in recycling endosomes. But in the case of cells expressing LRRK2 mutations, we see a sequestration of this internalized transferrin into these cytoplasmic LAMP2 positive vesicles. Now we can quantify the levels of internalized transferrin. This is an uptake assay as opposed to recycling. And in this case, we load the cells with transferrin, and we can take, again, by high throughput imaging, we can take ICC images and actually quantify the levels. So what we see is that with wild type LRRK2 or control cells, we reach a plateau of internalized transferrin after 10 to 15 minutes. But in the case of the mutants, that plateau is higher, which means that we have, but the rate of uptake is actually the same, but what we have after 5 or 10 minutes, we start overloading the system with transferrin, and we have accumulation of internalized transferrin, presumably due to lower recycling, because transferrin is probably pushed towards the lysosomal compartment as opposed to quickly being recycled back to the membrane. Now, to look at the association of these transferrin positive vesicles with lysosomes, we can do colocalization analysis, and you can see here with LRRK2 mutations, we see increased colocalization with lysosomes. And more importantly, to test whether this is a RAB8 mediated effect downstream of LRRK2 kinase activity, we knock down RAB8 in these cells, and you can see that the staining goes out completely. So it's a quite good antibody. And in this case, we have much lower sequestration of transferrin to these LRRK2 positive, LAMP2 positive vesicles. So what we effectively see is an effect mediated by LRRK2 kinase activity and the hyperphosphorylation of RAB8. Now, we have a dysregulation of transferrin recycling, and we see that transferrin is pushed towards lysosomes, and maybe we may have accumulation of internalized transferrin in cells expressing LRRK2 mutants. But what happens to the intercellular iron levels? Because transferrin is one of the ways that iron is internalized in the cells, right? So we did an ICP-MS looking at the levels of internalized iron. And what we see is that we see a turn of an increase in the R4041C cells and a significant increase in intercellular iron levels in the G2019S cells. And this, the one on the right, is now preliminary data because unfortunately we closed before we could finish the experiments, but we should be completing the picture pretty soon. In this case, we used a live stain dye where we can stain for ferrous iron internalizing cells. And what we see in this case, that in cells expressing R4041C, we see an increase in iron stain in these LRRK2 positive lysosomes. So this hints to a dysfunction of iron homeostasis mechanisms, and to see it to investigate in a more physiological kind of way, in a model that's more like linked to disease, we turn into microglia because we want to move away from cancer cells, as Luis was saying before. So in this case, we're looking at iPSC, human iPSC derived microglia, either wild type or carrying the G2019S mutation. And we're looking at microglia because it's one of the cells that express LRRK2, and one of the models where we think that LRRK2 plays a role and is also activated downstream of inflammation. So in this case, we stain for internalized transferrin and LAMP2, and what we see is that we see a partial colocalization of transferrin with lysosomes in cells treated with DMSO. Now, when we activate this microglia with LPS, we see a drop in colocalization of transferrin with lysosomes, and what we think that happens is that when cells are treated with LPS, when a microglia are activated in vitro and in vivo, you have this quick uptake of iron, which is a physiological response to proinflammatory stimuli. And this requires a fast uptake and recycling of transferrin. So we see a push of transferrin away from the lysosomal compartment and closer to the membrane for higher recycling. Now, in the case of cells carrying the G20S mutation, cells treated with LPS, they don't actually show the same shift towards higher, faster recycling. Now, these images were taken on super resolution AirScan platform. And one of the cool things that we can do with this is that we can use sophisticated software like Imaris and really try to reconstruct the 3D surfaces and look at the individual transferring and LAMP2 positive vesicles, compare them in terms of size and also in terms of like, how close they are to each other, how far away they are from the nucleus, if they're in distal and proximal sides and what have you. And here I'm just showing the example of the wild type and the LPS treated, just show you that what we can actually do with this technology. So when we do this kind of analysis, we can do the 3D rendering of the surfaces and look at the distribution of transferrin vesicles away from the nucleus. So in the case of the wild type, about 60% of these transferring vesicles are close to within 1 to 1.5 micrometers from the nucleus. And what we see is that when we treat cells with LPS, we see a wider distribution of transferring very closer to proximal sites. Now, when we look at the same kind of analysis in the G2019S carrying cells, they don't follow the same trend. And also, we can look at the mean distance to the nucleus that we have the transferring vesicles are closely packed to the nucleus in the G2019 cells, and also they are the average size of them are higher, are bigger. And so what we see is that we have a shift of these endogenous transferring vesicles towards the nucleus and in higher sizes and are associated with lysosomes in the G20 cells compared to the wild type. Now, if we want to take a step further and look at how this is, how can this translate into an in vivo PD model, we can use the G20S knock-in mice. So in this case, we compare wild type and GS knock-in mice and the response to intrastriatal LPS injections. So in this case, we inject LPS, and then 72 hours later, we can fix and stain with a pearl stain, which is has been around for many, many years. And it's basically a readout of iron deposition. So in this case, we're looking at the iron that's deposited in microglia following the LPS administration. So what we can do when we quantify the levels, we see that we have an increase in iron deposition in the G20S mice, which is higher to the wild type. And we look at which cells are actually responsible for this iron staining are predominantly microglia. So this again is a physiological uptake of iron is a physiological response to LPS, but we see that there's a divergence to pro-inflammatory conditions in terms of iron homeostasis in the context of the G2019S mutation. So, and we can take this a step further and see the packaging of iron and how iron is actually stored in these cells. And in this case, we looked at the ferritin heavy chain, which is part of the cage that stores iron in mammalian cells. In this case, ferritin is stained in red. And again, you can see a higher ferritin deposition and expression in G20S cells compared to the wild type. Now, if we look at the double staining, we can see that about 60% of cells that actually respond with increase of ferritin levels are EBA1 positive and therefore microglia. We didn't see much of an increase with transferrin, but then again, we don't necessarily think that our system will be fine-tuned to look at small changes of transferrin levels as opposed to intracellular localization changes. So this brings me to the model. So what we believe that happens under physiological conditions of wild-type LRRK2 expression is that RAB8 resides in between the early endosomes and the late endosomes, and the endocytic recycling compartment when mediates the recycling of the transferrin receptor back to the plasma membrane. Some of it will be targeted to lysosomes for degradation, and also lysosomes are one of the physiological storage places for compartments for iron in the cells. Now, when we move to the mutation, we see a shift of RAB8 towards late endosomes and lysosomes, and that brings together transferrin and it dysregulates transfer and recycling, and therefore we have, in conditions of proinflammatory stimuli, we have a shift towards lysosomes and upregulation of iron storage mechanisms. So with this, I would like to close and thank everybody in my previous lab. I'm not going to go through everybody, but you can see here that this has been an amazing experience, and it's a very good lab to join. Thank you so much.
Thank you, Louis and Adam, for those very excellent presentations. So we're going to move into our third speaker today, which is Dorien Roosen, who also worked with Adam and Louis at the NIH. And she will be talking about auxilin, clathrin trafficking, and Parkinson's disease. Go ahead, Dorien.
Okay. Can you see my screen? Okay. Okay, so hi everyone. I'm Dorien. I'm currently a postdoc at the Leibniz Research Institute in Berlin, where I'm studying membrane trafficking in the context of neurodevelopment. But 6 months ago, I graduated from my PhD in Mark Cooksson's lab at the National Institutes of Health and Patrick Lewis' lab at the University of Reading, where I studied membrane trafficking in Parkinson's disease. And so my PhD project was about characterizing mutations in auxilin that cause Parkinsonisms. So a lot of genes that have been associated with Parkinson's disease are involved with membrane trafficking in neurons. So for example, LRRK2, VPS35, RAB29 are involved with trafficking from the Golgi apparatus to endosomes. And we just heard from the talks from Luis and Adam as well that LRRK2 is also involved in lysosomal biology. And there's multiple other lysosomal proteins that have been associated with Parkinson's disease. And clathrin trafficking is a specialized form of vesicular trafficking. So clathrin trafficking happens at the plasma membrane for clathrin-mediated endocytosis. So it is involved with endocytosis of vesicles, which again then traffic to intracellular destinations. And clathrin trafficking is also important for the recycling of synaptic vesicles. And in addition to the plasma membrane, clathrin trafficking can also happen from the Golgi apparatus. And multiple genes that are associated with Parkinson's disease are also involved with clathrin trafficking, including auxilin, GUC, and synaptogenin 1. So the formation of a clathrin-coated vesicle starts with budding at the membrane. So it starts with the selection and the concentration of clathrin adapter proteins. And then as the vesicle grows and matures, it starts forming a clathrin coat around it. And when it's fully mature, it's pinched off the membrane. And at that point, synaptogenin is recruited to the vesicle to shed off the clathrin adapter proteins. And then after that, auxilin or the homologue of auxilin, GUC, is recruited to the vesicle, which takes care of the uncoating of the clathrin coat. And then the naked vesicle can travel to its destination compartment and fuse to deliver its contents. So auxilin is encoded by the gene DNA-JC6. And multiple recessive mutations in DNA-JC6 have been described to as a form of juvenile and early onset Parkinson's disease. So the age of onset of Parkinson's disease ranges between 8 and 45 years. And so in addition to the Parkinsonian motor phenotypes, a lot of patients also have atypical neurological phenotypes. Some of them suffer from seizures or other cognitive impairments. So far, 5 mutations have been described. There is 2 nonsense mutations that result in a premature stop codon. There is a synonymous mutation and also an exonic mutation, and both of those are within splice sites and result in decreased mRNA levels of auxilin. And then there is also one point mutation. Altogether, the genetics really point to loss of function mutations in the context of Parkinson's disease. And auxilin itself is a neuron-specific protein, but its homologue GUC, which has exactly the same function, is ubiquitously expressed. And GWAS studies have nominated GUC to be a risk factor for Parkinson's disease as well. So this point mutation is actually in a residue that's highly conserved across species. So we developed a novel mouse model for Parkinson's disease using CRISPR by mutating the equivalent mutation into the mouse genome. Auxilin expression in the brain as well, and you can see that auxilin expression is very high within dopaminergic neurons in the substantia nigra, which is the area that's impacted most in Parkinson's disease. We also had a look at auxilin protein levels, and we saw that auxilin protein levels are drastically decreased in the brain of these mice. There's also an increase rate, which you can see here by the deviation from the expected Mendelian inheritance, and mice also displayed slightly decreased birth weight. We then had a further look at neurological phenotypes. And as I mentioned before, some of the auxilin mutation carriers display seizures. And one of the things we observed during gait changes was that these mice exhibit seizures as well. So you can see that this is a 6-month-old mice. She's having muscle twitches, and then she freezes and starts drooling from the mouth. We also had a look at other motor phenotypes and saw motor impairment, for example, by the pole. So mice with auxilin mutation showed an increased time to turn and increased time to descend a vertical wooden pole. And when the mice were placed into a startle chamber, we also noticed that the mutant auxilin mice have an overall decreased amplitude of movement. So we did not observe neurodegeneration in this mouse model, but we did have a look at neuropathology using electron microscopy. And one of the things we observed was that the auxilin mutant mice have a decreased amount of synaptic vesicles in the presynaptic compartment and an increased number of clathrin-coated structures, which is indicated by the red arrows here. So this indicates impaired uncoating of clathrin-coated vesicles at the synapse. And another thing we observed in the striatum of the auxilin mutant mice were these large lipid protein aggregates. So to confirm this, we also did a lipid staining in slices, which is the dye for neutral lipids, and we looked at dopaminergic neurons in the substantia nigra. And what we saw was increased lipid content in dopaminergic neurons of mutant auxilin mice. They had less lipid particles, but the lipid particles were larger. So to get further insight into molecular mechanisms underlying these phenotypes and pathology, we did a sialic-based proteomic screen to identify the interactome of auxilin. We identified auxilin itself here as DNA JC6, multiple clathrin subunits, and clathrin adapter protein, which were basically the positive controls of the experiments. Now we did gene ontology analysis of the interactors of auxilin. And of course, we saw enrichment of clathrin-associated proteins, synaptic proteins. But what was surprising to us that we also saw a lot of an enrichment of Golgi-resident proteins, because auxilin so far had been thought of as a synaptic protein, and its role in clathrin trafficking at the Golgi apparatus had actually not been studied yet. So because of this, we had a look at the Golgi apparatus in mouse brain slices in mutant auxilin mice. And so the first thing we observed was that there was a significant increase of clathrin-coated vesicles around the Golgi apparatus in mutant auxilin mice, which again indicates impaired clathrin encoding. And then the other thing that we observed is that the mutant auxilin mice had a really swollen Golgi morphology. So in wild-type mice, the Golgi is observed as these dyed Golgi stacks. But in the mutant mice, you can see that it's very large and swollen. So we also looked at Golgi morphology then in using ARIE-scan confocal microscopy. So these are primary neurons derived from wild-type mice and mutant auxilin mice, and we stained for the endogenous Golgi markers for the cis, medial, and trans Golgi. And here again, you can see that the Golgi stacks are more separated because of the swelling of the Golgi morphology, and you can also observe that there's a decreased colocalization between the different markers. So then we had a closer look at the interactors that we identified with the Silex screen. And so I mentioned before that we recovered clathrin adapter proteins. So there's multiple clathrin adapter proteins that have very distinct subcellular localizations. So there's AP2, and AP2 is uniquely present at the plasma membrane of cargo selection and concentration at the plasma membrane. And then at the Golgi apparatus, there's multiple clathrin adapter proteins. So there's AP1, AP3, and then also the giga proteins, giga 1, 2, 3. And AP2 was a previously known interactor of auxilin, but in our Silex screen, we also identified giga 2, a Golgi-resident clathrin adapter protein as an interactor for auxilin. So we then wanted to confirm these interactions, of course. So we expressed either wild-type auxilin and also mutant auxilin to assess the effect of mutations on interactions. So either GFP wild-type auxilin or GFP mutant auxilin was expressed in cells. Then we did GFP immunoprecipitation. So we validated the interaction between adapter proteins AP2 and the new interactor GIGA2 as well. But mutations did not really seem to impact this interaction. Now we also had a look at interaction of auxilin with clathrin. So again, we expressed GFP wild-type auxilin or GFP mutant auxilin. And wild-type auxilin interacts very strongly with clathrin, but all of the mutations impair this interaction. So because clathrin interaction was impaired by all of the mutations, we had a look at the localization of mutant auxilin in neurons. So here we expressed either wild-type auxilin or mutant auxilin in primary neurons and co-stained for endogenous clathrin. And as you can see, even though that all the mutations lost interaction with clathrin, auxilin still colocalizes with clathrin vesicles. So this indicates that it's still capable of being recruited to these vesicles, but it's not capable of doing the clathrin encoding reaction. So then, as a summary, I'd like to finish with these slides, which presents a model for disease pathogenesis in auxilin mutation carriers. So in its normal state, clathrin trafficking in neurons can happen from the Golgi apparatus, where it takes care of cargo sorting to endosomes, but also of sorting of lysosomal proteins to the lysosome. And then clathrin trafficking also happens at the plasma membrane for clathrin-mediated endocytosis, also for delivery of cargo to intracellular destinations. But clathrin trafficking is also involved in recycling of synaptic vesicles. So we saw that auxilin is important for clathrin trafficking both at the Golgi apparatus and the synapse. and that pathogenic mutations in auxilin impair clathrin trafficking pathways. So impaired clathrin trafficking at the Golgi apparatus would result in decreased cargo sorting from the Golgi to endosome, but also impaired delivery of lysosomal proteins, which could result in these intracellular aggregates that we observed in the mice of auxilin mutant mice. And then impaired clathrin trafficking at the synaptic compartment could impair recycling of synaptic vesicles and ultimately result in the neurological phenotypes we observed and the decreased number of synaptic vesicles. So with that, I'd like to thank a lot of people that contributed to this project. So I won't have time to go through everyone individually, but I'd like to thank my PhD supervisors, Mark Cookson and Patrick Lewis, and everyone in the lab. I'd also like to mention the paper is in revision, but if it's already available on bioRxiv. And then I'd also like to thank the organizers for this initiative and all of you for attending. And then I think we're ready for some questions.
Yes, thank you, Doreen. Fascinating presentation. We'll kick off the Q&A session, starting with a question for Louis. Darren Hara said, great talk, fascinating stuff. Could you comment on how increased kinase activity with a G2019S mutation might be more pathogenic in this scenario? Increased LYTL.
Yeah, thanks for this question. It's really good. And if I'm super honest, I would say that I don't know. But for the sake of saying something else, I would say that the G2019S mutation basically increases everything, right? It increases the localization of TIP4, the RABs, and more tubulation. So the first thing that comes to mind is that, for example, we know that RAB, as an indirect effect, we know that RAB10 is required for mitophagy. So we know that mutations that hyperphosphorylate these RABs have a lower rate of mitophagy. So it is likely that if you have more GS pulling everything, trapping everything to the lysosome, it is likely that RAB10 is not able to then go to the mitochondria to just mediate this process. As an other way to think about this would be that you really need to solve, we really need to solve what this contact size between the vesicle and the lysosome really mean, right? Because as long as we don't know that as a direct effect of little, we just can't really go deep on this answer. It is likely that, or possible that this interchange of events can overwhelm the recipient lysosome, and that can be toxic. Or is it possible that somehow, somewhere, the vesicles actually leave the cell and get uptaken by a neuron, and that ends up being toxic? So there's a lot of things that we can do to really solve this, but I think it's a really interesting question.
Thank you. Now a question for Adamantios. Do you anticipate that you would see iron accumulation within the lysosome from a pathogenic increase in wild type LRRK2 activity when there's no mutation present?
So yeah, I think that's a very interesting question. So I should start by saying that iron deposition in the brain is a neuropathological feature of disease. In fact, we see iron deposition in a lot of neurodegenerative diseases. So there's definitely a link, even in the prodromal stage of a lot of neurodegenerative diseases with iron deposition that can lead to neurotoxicity. Now, in the light of LRRK2 activation and mutations, there is actually a study by Stéphane Leherisi at the CENIR in France, where they looked at the MRI images actually from idiopathic and Parkinson mutation carriers and LRRK2 mutation carriers. And what they see is that going from wild type from non-mutation carriers to mutation carriers of LRRK2 and Parkinson, you see a higher iron deposition even in the preclinical stages of Parkinson's disease. So there's definitely a link with LRRK2 that we can actually see in some early studies. Now, in terms of whether you can see increase of iron deposition without the mutations, well, there are studies which are really important, like Malutan's study, where they show that in idiopathic PD, you actually have higher levels of LRRK2. So there's definitely a way that I think that you can see R in deposition, even if in the light of no mutations, but higher levels can actually drive higher LRRK2 activity and downstream effects. Does that answer the question? That's a good
Excellent
question.
answer. So Dorien, your mouse model, were there any early onset pathology in that mouse model, or what age did you start to see this dysfunction, the phenotypes?
So we did see some early, like some perinatal pathologies as well. So they had lower birth weight, increased mortality rate. And we started seeing motor phenotypes at 6 months of age. So the data I showed today were in 1-year-old mice. The video of the seizure was in 6-month-old mice, but they do progressive motor impairment. So some of the phenotypes start to show at 6 months old and then progressively worsen as they age.
Thank you.
Thank you. We have a question from Sean Ferguson. So this is for Louis or Adamantios. What physiological circumstances would elicit lysosomal damage similar to what was triggered in the experiments with the LLOME? If we want to start with that beginning of that question.
Do I go? Okay. So this is, Sean, an excellent question as well. To be honest, I would say that, for example, uptake of, there's several ways you could go with that. It's been shown that in microglia, if microglia are able to eat too much myelin, actually triggers lysosomal membrane damage. So that's one. And another that is very interesting for the pathology is shown that, for example, tau aggregates or synuclein fibrils can also trigger lysosomal membrane damage. Now, whether it is exactly the same damage to trigger LRRK2 or not, that's something that would be really interesting to explore.
Thank you for that answer. So maybe we'll go into a final question for Dorien. So what do you think is causing the swelling of the Golgi in your mouse model? And is this a phenotype that you also see in the human brain?
So in terms of human neuropathology and auxilin mutation carriers, we don't know because there's no neuropathological data available. For the Golgi swelling, so one possibility could be that because of the impaired clathrin trafficking and the vesicles cannot fuse with their destination compartment and deliver their cargo, there could be a feedback control mechanism in place that signals back to the nucleus and the protein system, which would deliver stress on the Golgi apparatus as it's trying to compensate for this impairment in delivery. Yeah.
So maybe just a follow-up question on that regarding to patients. How prominent is this mutation in the PD population? How many PD cases?
Yeah, not prominent at all. So it's only been identified. So each mutation was identified in one single family only. So at the moment, I think there's between 5 and 10 patients living with auxilin mutations.
Okay. Well, thank you all for attending today, for those excellent presentations and for all the questions. Please let's continue this discussion on Twitter, on social media, or reach out to our speakers if you want to talk to them even more about this. Before finishing the session, I would like to talk about what's happening next week. We have a Tuesday session coming up where Lynn Crone, a PhD student at McGill University, will be talking about REM sleep behavior disorder genetics for early detection and subtyping of Parkinson's disease. And in that session, we will also have Karim Esat, who's a research scientist at Karolinska in Sweden, who will talk about loss of function mechanisms in amyloid pathologies. So thank you everybody for listening and have a great day.
Thanks a lot. Have a good day.
Thanks a lot, guys.
Bye.
See you later. Bye-bye.
Bye. Thank you.